All right, guys, so we'll pick up here with the muscular and cardiovascular system. So in fish, body muscles are comprised of a series of sort of zigzag-shaped myomeres. Each myomere constitutes one muscle segment, and the coordinated contractions of those zigzags produce waves. And this provides the main locomotion mechanism for fish. That wave moves posteriorly in the water, and the water adjacent to the fish accelerates backwards until it passes the caudal fin and produces thrust. So, in most fish, white muscles predominate. They can account for up to 90% of the entire body weight of a fish. White muscles have relatively thick fibers. They contain no fat. They're primarily used for anaerobic respiration. So, similar to the white meat that we see in the breast of chickens, they're associated with fast twitch muscle fibers and rely on anaerobic respiration and sort of quick, short acceleration. But those fish that swim long distances or most of the time and need to have adequate oxygen supply, like tuna and mackerel, rely more on red mussels. Red mussels make up a greater proportion of their body muscle mass. Red mussels differ in that they have thin diameter fibers, they contain fat and myoglobin, they're well vascularized, and they utilize aerobic respiration. Um, there are 6 different groups of fish that are known to possess electric organs, derived also from muscle fibers. Certain muscle masses in these fish are highly modified to produce and discharge electricity. Because these groups of fish are only remotely related, we know that electric organs actually evolved separately several times. 2 of the continents that they evolved on, South America and Africa, were separate by the time electric organs evolved in these fish. Some fish can discharge volts up to 500 volts, and others only emit weak electrical charges. Um, Electric organs have a few functions. They're used as a defense, they are used to stun prey, and they can also be used to scan the environment and locate enemies or prey, um, as well as being used for social communication. Moving on to the cardiovascular system. In fish, let's talk through the heart. So in fish, we have several parts, 5 sections of the heart, while the heart is comprised of 2 chambers. So the sinus venosus is the thin-walled sac that chiefly serves as a collecting chamber for venous blood that's received from all parts of the body. Blood flows through the sinus venosus into a large, thin-walled muscular sac, the atrium. From the atrium, blood enters the ventricle. The ventricle is a relatively large muscular cavity with heavy walls, and it is the primary pump for distributing blood anteriorly in the fish. A muscular tube, the conus arteriosus, connects with the ventral aorta and serves to moderate blood pressure. And in some fish, the conus is short, and its function is actually assumed by the bulbous arteriosus. So, again, although we define 5 different sort of parts of the heart with different functions, fish hearts are a 2-chambered organ. So in fish, all the blood must travel through at least 2 capillary systems, the gills and the tissues, before returning to the heart. So a drop of blood passes to the heart of a fish only one time during each circuit through the body. And after passing through the heart, the blood's carried to the gills for aeration, and then it's distributed really to all parts of the body. In most fish, all the blood flowing through the heart is venous blood. So that is in contrast to a drop of blood in amphibians, reptiles, birds, and mammals that must pass through the heart twice during any single circuit of the body. So the figure on the right bottom shows the single flow plan of the fish as opposed to the resulting double flow circulatory pattern that we see in other vertebrates in our tetrapods. You know, although we think of fish as ectodermic, there is some degree of endothermy present in some fish species, and particularly in shark species. So in certain oceanic fish, including sharks and mackerels, tuna, billfish, we see a endothermy that really requires a high aerobic capacity. So again, relating back to the red muscle fibers that we see in some of those types of fish. And so it depends on one, high aerobic capacity, and two, some reduction of heat loss in those fish. And there's a surface area to volume ratio that helps with minimizing heat loss in the aquatic environment. So they're able to retain some of that metabolic heat by way of a countercurrent exchange system. And so in several fish, we see them using just a moderate degree of endothermy to warm the brain. Billfish show this kind of cranial endothermy and will warm only the brain and eyes. They do that by passing blood through the vessels around the eyes. And they think that that kind of cranial endothermy may have allowed for some of these fish species to expand their range into cooler waters. We'll hear more about the use of countercurrent exchange for heat retention in other vertebrate species. So I encourage you to look at the figure in some of your books. The concept, though, is that the coolest blood is coming into contact with only moderately warmer blood. Then this allows for the maximum exchange of heat between blood flowing out of the cranial region and blood flowing into the cranial region by trying to maximize the differential between blood returning and blood leaving. Hard to describe without a better graphic. So spend a little time with countercurrent exchange mechanisms, because we will talk more about that. Two more systems: respiratory system and digestive system. So respiratory systems in fish, we know that the gills function primarily for respiration. They're ventilated by unidirectional flow of water created either by active brachial pumping or passively by simply opening their mouth while they're swimming forward. This system consists of several major gill arches on each side of the head, with 2 rows of gill filaments extending from each gill arch. Each filament consists of rows of densely packed flat lamellae. Gill rakers are present to protect the gills and direct food towards the esophagus. Within the gills and at the site of gas exchange, water enters the pharynx through the mouth or spiracles and passes into the gill chambers, where it really bathes the gill surface. Water flows front to rear through the slits between the lamellae. Gas exchange takes place there, the site of lamellae. At the same time, blood flows through the capillaries in the opposite direction. And again, countercurrent flow really helps to increase the efficiency of oxygen absorption from water. So water can leave the gills of bony fish with as much as 80 to 90% of its initial oxygen content removed. That's in contrast to the air that mammals breathe, which only removes about 25% of the oxygen present. So it's the efficiency of the fish system is impressive. Now remember that fish also live in an environment with only about 1/20th or 1/30th of the amount of oxygen that we have in air. So that efficiency is really needed. The digestive system, we'll talk through now. Fish use a variety of different strategies to secure food to meet their metabolic needs. We have as many different strategies out there as we do have types of fish, it seems. So this includes filter feeders and those that feed on plankton, herbivores that feed on plant materials, detritivores that consume partially decomposed organic matter, carnivores that feed on animal material, and omnivores that feed on a variety of plant and animal material. The stomach's a muscular organ at the end of the esophagus that really is just an enlargement of the esophageal system. It secretes enzymes and serves as a temporary storage site for food as it's moved through the digestive tract. This distinction between the esophagus and stomach really can be poorly demarcated in fish and hard to see. Little blind pouches known as pyloric cecae are found near the junction of the stomach and the intestines in many species, and their primary function is to help with fat absorption in those species that have them. And the digestive tract is a relatively straight tube. Predatory fish have a relatively shorter intestines than we see in herbivorous fish, really because carnivorous fish have an easier time digesting their food than a plant-based diet. Some chondrichthyes have a specialized structure called a spiral valve within their digestive system. And we haven't really discussed the cloaca yet in this class. We do see a common vent in some fish species that we'll also see when we discuss birds and reptiles. So the cloaca is a common chamber at the end of the digestive tract. It receives the colon, the urinary tract, and the genital tract, and opens into a single vent. Other fish species have separate openings for each of these systems. And swim bladders. Swim bladders originated as modified paired pharyngeal pouches. They originally evolved as organs for respiration in some species, and then some species evolved those into true lungs. In most modern fish, they serve a different function. So, because the density of animal tissue is greater than the density of water-dwelling species tend to sink. They can counteract this either by actively swimming or passively by the development of a gas-filled swim bladder. So the function of the swim bladder really is to serve as a hydrostatic organ that regulates buoyancy. It's filled with oxygen, carbon dioxide, and nitrogen. Those are in higher concentrations than we would be expected just through air breathing. The source of the gases is normally extracted from the blood, but some fish do gulp air to add to their swim bladders. A lot of bottom-dwelling species have lost their swim bladders as an adaptation to that lifestyle. And in order to maintain buoyancy in cartilaginous fish, who also lack swim bladders, sharks never stop swimming. So that's the alternate plan to maintain buoyancy. Swim bladders can serve other functions. They can be involved with hearing and creating sound. So some fish use them for communication. The organs in fish are fascinating and also diverse. So we'll talk through a few of these sort of quickly. They possess a neuromass system. So this consists of fluid-filled pits and ampullae and canals. That system is responsible primarily for receiving mechanical stimulation from water movement and sound waves. This enables fish to avoid potential predators, to orient themselves, to locate food, and also to participate in schooling. In some fish, the system is sensitive to even minute electrical currents in the water. And by sensing the electric field, fish can actually use the neuromass system to see in a way. It creates shadows that can be discerned and give information about the size and shape and movement and electrical properties of other organisms around that fish. So that system is important for schooling fish. When schooling, using the neuromass system, fish are able to monitor the swimming speed and direction and distance of their neighboring fish. And that allows the school as a whole really to execute some really complicated maneuvers that require individuals to respond really quickly and change directions and speed and direction. So ears in fish function a little different from our ears. All fish have a series of tubes and sacs and membranous labyrinth of the inner ear that's embedded in the auditory region of the skull. And ears are mostly involved in maintaining static equilibrium and dynamic equilibrium. And in just a few fish, the ears develop the accessory function of receiving sound transmission. In some cases, by being linked to their swim bladders. Eyes, we do see paired eyes that really stay relatively constant through all vertebrates. Many fish have two lateral eyes that serve as their primary receptors of light. Most of these eyes are symmetrical. Flounder are an interesting example of asymmetrical. The penile and parapenile organs are also light sensing organs developed by a pair of sacs on the top of the head, but they do not create full images. In lamprey and some larval fish, they're the primary organ for light detection. But in other adult fish, they seem to be responsible for tracking photoperiod and solar radiation. So in most adult fish, they're thought to function for tracking seasonal changes in circadian rhythms of daily and seasonal cycles. Olfaction and taste. Olfactory cells can detect extremely small concentrations of chemicals dissolved in water. Olfactory clues can be shown to be extremely important in fish like salmon in helping them locate natal streams. And we talked about the fact that they have seen the same thing with lamprey and have used that in controlling lamprey populations in the Great Lakes. Sharks possess an extremely acute sense of smell. Taste cells are located on the roof, the sidewalls, and the floor of the oral cavity. The number of taste buds can vary greatly from one region of the body to another, and certain bottom feeders and scavengers like catfish actually have these kind of chemoreceptors distributed all over their body surface. And I've got a fun link here to another article that you guys might enjoy. The vertebrate endocrine system, I'm not going to go into a ton of detail about this. The book does give a detailed description of the endocrine system and notes that the system is similar enough across vertebrate species that this is the primary place where it describes the function of the endocrine system. But if we went into detail, I could spend a full hour talking about the endocrine system. So take some time to read this section, understand the important functions of the pituitary gland, the thyroid gland, the adrenal glands, the pancreas, and the gonads in producing hormones that regulate our growth, reproduction, parenting behaviors, and processing of sugar and salts and other regulatory functions. The urogenital system we'll talk briefly about. So I'm mostly going to concentrate this on osmotic regulation. So in a lot of marine fish, they're required to regulate an osmotic concentration of about 1/4 to 1/3 of that of the seawater around them. So they really have to have a dialed strategy that allows them to excrete excess salt from their system as their body tries to constantly take in salt to reach equilibrium with its environment. Sharks maintain that osmotic equilibrium by actually retaining large amounts of urea in their body fluids. And this raises the osmotic concentration in their blood to close to equal to that of seawater. Fish that migrate between freshwater and saltwater, such as androgynous fish like salmon, have to deal with going from a hypotonic environment to a hypertonic environment. And there's a suite of strategies to deal with that. In some cases, that osmotic regulation is thought to be controlled by hormones. Fish actively absorb sodium chloride in some cases to flush out excess water. The kidneys can produce a really dilute urine by reabsorbing some of that sodium chloride. And then marine fish alternately drink seawater and replace water lost osmotically with excess salt while the excess salt is transported out through the gills. And both larval fish and hagfish use a pronephritic kidney, while adult fish have developed a functional kidney. And what serves as the functional kidney in many adult fish actually serves as the embryonic kidney in reptiles, birds, and mammals. So just an interesting staging of the evolution of the kidney through those different taxa. Growth and reproduction in fish is as varied as these other systems. So some of the strategies for reproduction include the fact that we see sexual dimorphism. We'll talk about that on the next, or I've got a picture of that on the next slide. So in many adult females tend to be larger than adult males. And we know that sexual dimorphism is particularly evident in those fishes that bear live young. In others, especially species where the males compete for the females, the males tend to be the larger sex. And sexual dimorphism might involve special structures We see development of tubercles on the heads or changes in the scales of some of the male fish during the breeding season. And males of some species become brightly colored during the breeding season. And some fish engage in pretty elaborate courtship behaviors. For example, the males of the three-spined stickleback fish construct a tube-like nest and then use a really complex courtship ritual to entice the females to enter their nest. Siamese fighting fish build a bubble nest as part of their courtship behavior, and females expel their eggs close to that nest, and then the male blows them towards that floating nest mass, and where they unite with the bubbles, and then the male continues to guard that until the larva hatch. Most female bony fish release eggs into the water where the sperm's been deposited. Other fish hide their eggs in gravel or rocks or build nests in which eggs are buried. We talked about the fact that lamprey excavate redds, and we see that same behavior in salmon and trout. And then internal fertilization occurs in most cartilaginous fish. Lots of fish with internal fertilization carry the embryos until the time of birth, but some fish protect their eggs or offspring by carrying them in their mouths. This is known as mouth brooding, and been seen in catfish and cichlids and the cardinal fish. As in this picture, male seahorses have brood pouches and are marsupial and hold their young in a pouch in the belly in which the female deposit the eggs. We see hermaphrodity in some species, and we see extreme variation in the number of eggs that a single individual can lay. There may be just a handful, or it's up to 5 million in other species. Growth and development in fish is also varied and really separated between oviparity and viviparity strategies. So in some viviparous species, embryos use their yolk sac supply until it's completed, but then stay in situ in uterus and hatch within the uterus and stay there in their larval stage. In some cases, fish continue to ovulate, and the young will ingest the eggs that are released through ovulation. In others, a species of rays, once they run out of their yolk sac, they secrete a nourishing fluid into the uterus known as uterine milk that allows the young to continue to feed while they're there. Generally, development of fish is thought to move through 4 stages from embryo to larva to juvenile to adult. The larval stage begins when fish are able to catch their own food. During that time, the skeleton and the other organ systems really develop more fully. In the juvenile stage, organ systems grow considerably, and fish are considered mature adults once they are reproducing and have mature gonads. So that time to reach sexual maturity can range from a few weeks to several years. So I've got a couple of slides here showing sexual dimorphism in fish, which can be extreme, and also these varied strategies for reproduction. And as with other chapters, just a few questions picked from the end of the book for you guys to do some review with. And that is it. It was a long one. All right, talk to you soon.